Do you ever wonder why learning chemistry is important? It's a springboard or jumping off point for many other sciences. Take the study of climate history, called paleoclimatology, for example. How do scientists who study paleoclimate make correlations between glacial and interglacial periods and ocean currents? Through chemistry. It starts on board a really special ship called the IODP Resolution that can drill into the seafloor and recover sediments and rocks from millions of years ago. On this expedition, scientists wanted to explore the bottom of the North Atlantic. The drill crew put a bit attached to a drill pipe through a hole or moon pool in the bottom of the ship in a process called tripping. When the ocean floor is reached, sometimes more than 2 kilometers under the surface of the water, drilling can begin. A 10-meter-long plastic core liner is fed into a metal core barrel and dropped down to just above the drill bit. And as the bit goes into the sediments and rocks, the core fills the core liner. Once it's pulled back up, the full core liner is pulled from the metal core barrel and it's brought to the core laboratory to be examined by the scientists on board. After being X-rayed and analyzed, the sections of core are split down the middle into long halves so the scientists can see what's inside. The scientists on board the IODP Resolution come from all over the world to work and study these cores. They work in the core laboratory on board the ship in the middle of the ocean, making preliminary observations and measurements. The cores are then put in the ship's cold storage for the rest of the expedition. After the expedition, the cores are sent to their permanent home, called a core repository. These cores went to Marem, a marine research facility in Bremen, Germany. After reviewing the preliminary data, each scientist decided which parts of the cores they wanted to study for their research. Once decided, they all got together in Germany to take samples from the cores. During this week-long sample party, the scientists got to discuss their upcoming research and collaborate on objectives while collecting almost 20,000 samples. These samples were sealed into labeled baggies and sent all over the world to their research facilities for further study. Some of the scientists, including Dr. Sidney Hemming and PhD student Claire Jasper at the Lamont Doherty Earth Observatory, are using their samples to study the changing ocean currents in the North Atlantic during glacial and interglacial periods. An understanding of the paleoclimate gives scientists data to clarify the implications of a warming ocean on the ocean currents that start in the cold North Atlantic waters and circulate throughout the world. I went to the Lamont Doherty Earth Observatory to learn more about how chemistry can help discover the Earth's secrets. Here, the labeled samples that were taken in Germany are frozen and then freeze-dried before they are analyzed. After being weighed, samples are agitated with distilled water so the clumps of sediment break apart and are uniform. This is important to free any small rocks or pebbles from the sediment, and it will help them sieve out the smaller components that they are not interested in right now. A machine helps Claire sieve the sediment into 2 parts, one that has pieces smaller than 64 microns and another with the larger pieces. The larger pieces will be put into an oven to dry. Both Claire and Dr. Hemming are interested in the larger fractions because they contain the clues to both age and origin of the sediments. Even the larger fractions need to be viewed under magnification. Here, Anna prepares a sample for us to see. She first uses a 160 micron sieve to ensure that the dust and smaller fragments are gone. This sample came from approximately 125 to 135 meters below the ocean floor of the North Atlantic. The sample is carefully put onto a viewing tray. Claire and her assistants will separate out the tiny shells of organisms called foraminifera from the abiotic components of each sample. To do this, a researcher uses a tiny paintbrush to pick out the almost microscopic shells under a dissecting microscope. Anna is looking for a specific species of benthic or bottom-dwelling foraminifera in the Cibicidoides genus. These organisms will be used for a type of chemical analysis called discrete isotope sampling, which, with other processes, can indicate whether or not the Earth was in a glacial or interglacial period when the sediment accumulated. The shells of the foraminifera are made from calcium carbonate. CaCO3, so incorporate carbon and oxygen from the surrounding water. The foraminifera that live on the bottom of the ocean are the most reliable proxy or measurement because the water down there is more uniform in temperature and salinity compared to the surface water. Also, because the North Atlantic is where the cold, salty, deep ocean current begins, the forams that live on the bottom there are some of the first organisms to record changes in ocean circulation in their skeletons. The changes in climate are recorded in oxygen isotopes within the calcium carbonate shells. Two forms of oxygen are studied in the lab, oxygen-16, which is the most abundant form, and a heavier isotope, oxygen-18, which is more rare. The ratio between these two isotopes changes as the climate changes. This is due to the fact that oxygen-18 does not evaporate as readily as oxygen-16. This means that when the evaporated ocean water is turned into snow that falls and gets incorporated into glaciers, the oxygen-18 concentration in the ocean increases because all of the oxygen-16 that evaporated is not coming back into the ocean, as it would during a warmer period where the snow melted or the water fell as rain. How do scientists get to the oxygen? They access it through a chemical reaction that releases CO2 from the CaCO3 in the shells. In the discrete isotope lab at the Lamont Doherty Earth Observatory, tiny samples of shells weighing about 25 micrograms or 2.5 times 10 to the negative 5 grams are put in small glass tubes in a carbonate device. The computer software will then load the tubes and add 3 drops of phosphoric acid to the sample. This creates an acid-base reaction where CO2 is released. The reaction is completed at a constant 70 degrees Celsius, which helps ensure that the reaction will proceed at the same rate for each and every sample. The product that the scientist wants to study is the CO2 vapor. This gets separated out from all of the mixture gases in the tube by freezing the sample at negative 160 degrees Celsius and turning the gases into solids. Because CO2 has a lower melting point than the other molecules in the mixture, it can be separated out using temperature. The CO2 separated out is ultimately warmed to 40 degrees Celsius and compared to a reference sample, which is one that has a known isotope ratio. Remember that the isotopes of oxygen have different weights, so can be distinguished by mass. Once the ratio of oxygen isotopes is compared to the glacial interglacial models, the climate during the time of the sediment deposit is revealed. The mineral materials in the sample can then be analyzed for other isotopes, which will help identify whether or not they came from Iceland or from Europe. This data will show how the circulations of currents change during times of glaciation.